Welcome to the second webinar of our 2017 Great Basin Landscape Conservation Cooperative webinar series. I'm Matt Germino, the Research Landscape Ecologist for the LCC. For those of you who may be new to the LCC, we are a partnership among public and private groups working to meet large-scale conservation challenges across the five-state Great Basin region. We have supported a variety of conservation projects on high-priority topics. And this series highlights a few of those projects, plus more. And we hope the webinars can provide a platform for discussing research and its applications to the Great Basin. We will keep today's combined presentations relatively short, about 35 or 40 minutes total, and we'll have ample time after for questions and discussion. And that will come after the last presenter. And since there's a lot of people on the webinar, we ask that you in the audience type your questions into the webinar tool as your questions come up. We'll read out your questions for the speakers again after the last presentation. And I want to note today's webinar will be recorded and it will be available on our website. So here's how to enter your questions. On your screen, you'll notice a small collapsed control panel in the upper right-hand corner. And by clicking on the small arrow button, you can expand the control panel. And in this expanded control panel, you can send in a question and just simply type it into the chat area and press send. And your questions will be read out after the last presenter, as I noted. So speakers, please remember to tell me when to advance your slides. And just so everybody knows, if we happen to have an internet or phone hitch, which seems to occur frequently in our office, then Emma at Enviro Issues will take control of the presentation and keep it going. So here's some introductory points to help us get started. And I think most of you already know that exotic annual grasses like cheatgrass and Medusahead have contributed in a significant way to the degradation of Great Basin ecosystems, and they pose a threat to much more acreage, and control measures are needed. These exotic annuals have altered the environment, particularly fire regimes, to the benefit of the exotic annuals and to the detriment of desirable native perennials. And feedbacks like the exotic annual fire cycle combine with variable weather to create one of the most significant and challenging invasive plant and restoration situations worldwide, right here in the Great Basin. Managers and scientists are hopeful that strains of a bacterium called Pseudomonas Fluorescens that has been selected for its weed-suppressive properties in laboratory, greenhouse, and wheat field trials will also be able to selectively inhibit the root growth of these exotic annual weeds in more complex rangelands. Weed suppressive bacteria are now commercially available in a number of states, and as you learned today, they've been applied already to tens of thousands of acres. But the results are variable and largely unvetted and unpublished. And so much remains to be understood about when, if, and then when, where, and why these bacteria are or are not effective on exotic annuals. And I want to point out that the proper way to introduce a control measure for exotic plants, such as is used in herbicides, is to first test that they're effective in doing the job. In other words, that they decrease the targeted exotic species with minimal impacts to other biota. And an important take-home message, I'll tell you right now, is that weed suppressive bacteria are not well tested, and that's in spite of their availability. And in a very real sense, the cart has been put before the horse. This is not the most efficient way to proceed, but fortunately, a small number of individuals have risen up to try to correct the situation and get the horsepower of scientific guidelines in front of, or at least alongside, the cart. And we're going to hear from some of these individuals today. So here's a few more points to consider. What do we need for a good control agent? As I mentioned, we want to know that the agent has been tested and proven to impact the target species with minimal impact to other taxa. But significantly, in the case of exotic annual grasses, they are impacting many millions of acres in the western U.S. And in order for a tool to be viable, it has to be cost effective. If it costs $100 or $200 an acre to apply, it probably won't be used at the scale necessary to have an impact. So that's significant. Also related to cost effectiveness, the tool probably will need to be able to be integrated with existing treatment delivery systems, such as able to be sprayed from aircraft or used with rangeland drill seeding, for example. There's a lot of practical issues. And in addition to these basic elements, people have also hoped that these weed suppressive bacteria, which by the way, we'll use WSB in many of the presentations as an abbreviation, we hope that perhaps they might be more acceptable. The species, Pseudomonas Fluorescens, is widespread and considered native to all of the subject soils, although the strains that are used may or may not be native. So it has a natural-ish type of element to it, and perhaps it would be more acceptable to society than some of the stigmas that we hear about with herbicide chemicals. The bacteria, as currently recommended, are applied in a very low density. And the idea is to get those bacterial populations to grow in the environment over the first year following application. And so years 2, 3, and 4 after application, we hope to see effects. And then by year 5, the concept or some of the assertions that we've heard is that the bacteria fades away in the soil and is no longer present. And I've heard that this is exciting to land managers because they feel that they need a tool that can provide a temporal bridge between the short-term action of herbicides, which is about a 1 to 2 year period, and the long-term benefits of the recovery of bunch grasses. They provide competitive pressure against invasion of exotic annuals. But bunch grasses take usually at least several, usually 5 to sometimes 10 or more years to recover. So there's a large time gap between herbicides and bunch grasses. Perhaps weed suppressive bacteria could fill that niche if they work. And the other thing that we hear from land managers is that tools that can be used in areas that are not already completely dominated by the exotic annuals are desirable. Tools that can be applied to sagebrush stands that have an incipient new invasion of exotics. A tool that can work there without knocking out natives would be very desirable. Now, one other consideration here is that we are, in a way, tampering with a black box. Having personally studied the microbial communities of sagebrush steppe and other rangeland environments, I know that there's not a lot of publications. There hasn't been a lot of study, and yet here we are talking about manipulating the bacteria in the soils of these environments. Some obvious risks exist there. Let's keep in mind also, though, that any tool we use on the landscape will bear some risk. We can't move forward without taking risk. But pathogens in general aren't really as known so well in these semi-arid environments as they are in like tallgrass prairie or forest environments that are more mesic and more hospitable to microbes. In sagebrush steppe, it's a cold desert. It's extremely cold in the winter and very hot, especially on the soil surface where bacteria would be applied in these tools. It's difficult to grow microbes. Soils in sagebrush steppe and related ecoregions are relatively sterile. They have a very low, small amount of microbes. And one of the best and few well-known situations of pathogenic microbes comes from Susan Meyer's research on the pathogenic fungi. You can see a photo of some of them here that attack cheatgrass. It turns out that there's a suite of 5 different species, and Susan and her colleagues have done a really nice job of characterizing the basic biology and ecology of these fungi. At this point, it doesn't appear that there's a lot of promise to do things like spray the spores of these fungi across landscapes and have desirable effects. But I would encourage everybody to take a look at what does understanding these soil microbes entail. Her research really helps us understand that. Okay, that's enough for me. Now let's hear from our speakers. The first of which will be Dave Pyke from the USGS in Oregon. Dave has worked for many years on many aspects of rangeland ecology and sagebrush steppe, and he's very well poised to give us a broader view of how we deal with controlling exotics, some bigger points to consider. Richard Lee is the IPM specialist for the Bureau of Land Management. He's stationed in Colorado. And Rick has done due diligence and a lot of great work to everybody's benefit on trying to find out what is available for these weed-suppressive bacterial strains and what is their status in terms of permitting. Rick has also been a leader in the BLM for generating a west-wide set of demonstration plots. In the Fish and Wildlife Service, Mike Gregg has been engaged for many years now with researchers who are doing the early testing on weed-suppressive bacteria. And he also initiated some early coordination events that I think had high impact on assessing these weed-suppressive bacteria. Jay Mangold is a outreach professor up at Montana State University who's also worked in the Great Basin testing different control measures, including herbicides and now these bacteria. And I think it's useful here in the Great Basin to take a look at what's occurring in neighboring ecoregions to compare and contrast. That's a useful exercise for us. And so Jane's going to present to us on what's happening up in Montana. And then lastly, Bren Lazarus is a postdoctoral associate in my lab here in Boise. And she has been dedicating her time to scientific studies of the effectiveness of weed-suppressive bacteria. And recently, with some support from the BLM, she is also helping us collate all of the different management trials and scientific studies occurring in the Western US on this bacteria. And there's some great insights to be had from her efforts. Okay, now I'm going to shift to Dave Pyke's presentation. So let's have Dave on the speaker.
Thanks, Matt. I'm going to start off by just, I've got 3 examples here of cheatgrass, Medusahead, and red brome. But those are just merely examples. We have other plant species that certainly fit this category of plant species that would likely be susceptible to weed-suppressive bacteria. But the invasive annual grasses, they clearly have impacted our ecosystems through changes in fire regimes, through changes in ecosystem processes, and in competing with the native plant seedlings as they try and establish. The worst individuals likely arrived in the United States in the form of seed contaminants with wheat and barley seed that was brought in the late 1800s and eventually spread along transportation routes, and they eventually filled voids that were available, as especially within the Intermountain West, where we had a considerable amount of overgrazing in the early 1900s. And then along with that, we had abandoned homesteads that started cropping up in the early 1900s, and that left lands that were devoid of vegetation. And as a consequence, then we had these plant species that were quickly spreading through areas, and cheatgrass was clearly the one that spread the quickest, but Medusahead has continued to spread, and even to this day is spreading into new environments. Next slide. So when we talk about control options, then for these worst species, and I categorize this as we're talking about the plant species that are already dominating landscapes, as opposed to new invasive species that are coming into locations. We can put the control options into three categories, and this is just a hypothetical curve, but one on the y-axis that is going from plant composition of 0 at the bottom to completely dominating at the top. And then as that changes in terms of the relationship between the invasive plant species relative to the native plant species within a community, our options for doing things with those plant species may change. And so based on the dominance of the invasive species, when the invasive plant is low and native perennials dominate, managers might consider managing for actual coexistence with the invasive species and making sure that they keep the perennials in the dominant category and that the invasive species are retaining just a minor component within the plant community. In contrast to this, this would be if we had a invasive species that was new to the location, this is where we would do our rapid response type of application for the invasive species. But in the case of most of these invasive annual grasses, they already are spread to considerable landscapes and many places dominate. As the invasives begin to approach the composition of the perennial plants, as we start to move up in that in the middle portion of that graph, the more of a passive management changes or selective treatments may impact the invasive plant species, and we can maintain or increase the perennial plants would be the goal that we would try and shoot for under that kind of situation. But when the invasive clearly dominates the landscape, as the further right in that graph shows, in those particular cases, restoration of the native plant community would be necessary in order to try and get any kind of control on the invasive species, and that's going to generally require killing most of the plants in the area. Everybody on Usgs. Next slide.
Can we have everybody go on mute, please? Except for Dave.
All right, well, it sounds like we got it. Okay, so let's look at some of these options really quickly. The do nothing or coexistence option, in that particular case, we're going to need to track the factors that are known to be related to the increases of the invasive species. So tracking things like bare soil, the amount of perennial plant cover, and the biological soil crusts on an area might be important for understanding whether you would expect to see invasive plants beginning to increase in a location. Also, the distance among the perennial plants, largely because that relates to the density of the perennial plants, might be important to monitor as well. As these changes then occur, then management changes might be considered. So if you were to detect changes, then trying to change whatever the current management on that area to be able to favor the perennial plants and the biological soil crust and keep the bare soil to a minimum. Next slide. At the other extreme of the spectrum, then, when the invasive annual grasses dominate those areas, then killing all of the plants that are on that site and going into a restoration of perennials on that location is oftentimes one of the better options to choose. There are a whole suite of herbicides that are available that might be able to provide this as a control for the invasive species, and then moving in with replanting and revegetating with the perennial plants in that community, so that we end up with a dominant perennial plant community on that landscape. And the restoration is largely necessary on those areas because the perennial plant communities generally do not have a sustainable seed bank that could be, that will recover after control of the invasive. Next slide. Then looking at that central portion of that curve for the passive or selective options, when we're found in between those two extremes, we prefer not to kill everything on those locations because we do have plants that are perennial plants that we'd like to be able to keep, but we would like to do some sort of a management treatment that would favor the perennials at the expense of the invasive annuals. And this is where a lot of the research is currently focused. Right now, there is a number of research projects that are looking at targeted grazing as a potential treatment that might favor the perennial grasses and actually reduce the invasive annuals, but the jury's still out on that. Relative selective herbicides may also provide reductions in invasive species, and those perennial plants that may be able to survive the herbicide could potentially increase, although there is some research that's showing that there's temporary harm to the perennial plants as well. So basically the jury's still out on both of those kinds of techniques. And so we're really entering into an era then of examining things like weed suppressive bacteria because of the large potential for impacting the invasive plant while at the same time not impacting the native perennials. And so that's been the attractiveness of moving in that direction. Next slide, and I'll either pass it over to Matt or to Richard. I'm not sure who's going.
Thanks, Dave. Richard, you're up.
Thanks, Matt. We're all aware of the tremendous impact downy brome or cheatgrass has here in the West, ranging from altering fire frequency to affecting sage-grouse habitat. Any new management option for this Bromus species would be of interest to state, private, and local landowners and managers. Next slide. In 1989, Dr. Ann Kennedy, USDA ARS, Washington State University, discovered and isolated a strain of Pseudomonas Fluorescens, which had activity on downy brome, Medusahead rye, jointed goatgrass, and to a lesser extent, Japanese brome. Initially considered a biocontrol agent, once identified as having pesticidal activity, the product became a biopesticide, with any further development for commercial use would fall under the EPA registration process for biopesticides. As with traditional herbicides, EPA has an outline process for the registration of biopesticides involving the documentation of the toxicology and the behavior and fate of the proposed active ingredient. The three strains of Pseudomonas Fluorescens, the D7, ACK55, and the MB906 that we're interested in today are not the first strains of Pseudomonas Fluorescens to have pesticidal activity. The CL145A strain is the active ingredient in the molluscicide Zequinox, used to manage zebra and quagga mussels. But these are the first soil bacterium identified as having selective herbicidal activity. The D7 strain was the first strain identified by Dr. Kennedy and became the first one analyzed for consideration as a biopesticide. Following the procedure outlined by EPA, the registration package for the D7 strain was submitted to the EPA in October of 2013 with a proposed trade name of D7 for their review and analysis. 10 months later, on August 29th, 2014, EPA granted the registration held by Verdesian Life Sciences of D7 for the, quote, suppression of downy brome, cheatgrass, Medusahead, Japanese brome, and jointed goatgrass on wheat, barley, triticale, oats, and rangeland, unquote. In 2001, Dr. Kennedy isolated the second strain, ACK55, which, like the D7 strain, has activity on downy brome, Medusahead rye, and jointed goatgrass, with a mechanism of activity associated with the disruption of root cell membranes, resulting in the stunting of roots of susceptible species. In September of 2015, USDA ARS, who holds the registration, submitted the registration package to the EPA for their review. Next slide. The following spring, of 2016, EPA notified the USDA ARS that they had identified some data deficiencies in the registration package, requiring additional studies in order to address the data gaps. In a recent email, it's noted that all of the studies requested by the EPA should be completed by the end of August this year, with an anticipated time frame for EPA's review and analysis of the data, resulting in a possible response from EPA early in 2018. The final strain of Pseudomonas fluorescens, the MB906, is the active ingredient of the product MB906, manufactured by BioWest Ag Solutions, which, unlike the D7 and the ACK55 strain of Pseudomonas fluorescens, has not gone through the EPA registration process, documenting the toxicology and behavior of this particular strain. Next slide. Following the registration, I received a call from Verdesian Life Sciences inquiring regarding the Bureau's interest in establishing some research and demonstration sites involving the D7 product. The BLM is not the research branch of the Department of the Interior, but we are interested in evaluating and gaining experience with new technology. Working with the interested field offices, sites were established in 7 states involving 11 field offices. Following the process identified by the Bureau for Research and Demonstration Products, as outlined in the 2007 Programmatic Veg EIS and IMs and informational bulletins, these projects were established. 8 studies were established in 7 field offices in the late fall, early winter of 2015. 7 additional studies were established in 2 field offices early in the spring of 2016. Later in the fall of 16, 2 studies were established in 2 field offices, and then in the spring of this year, an additional study was established. Of the 18 studies in the 7 states, it would appear that maybe the site in New Mexico is probably the southern edge of the efficacy of this particular strain of Pseudomonas fluorescens. Now, the summary of the studies that went out, the majority involved the broadcast application of the D7 alone. There were a couple which incorporated the taint mix application of imazapic, with one application of D7 being applied following an earlier application of the imazapic at 7 fluid ounces of plateau. Additionally, there were a couple of studies involving coating of seed and either broadcasting or drilling the seed. Next study, next slide. What we expect to see with the application of these strains concerning the management of downy brome is not going to be the same process we expect from the application of a herbicide active ingredient such as imazapic, rimsulfuron, or indaziflam, as Matt's already talked about. This takes time for the bacteria to build up in population, sufficient to provide the desired reduction in vigor and population of the downy brome, which would explain why some of the treatments involved a taint mixed application with the imazapic. Next slide. Regarding the immediate future and different strains of the Pseudomonas fluorescens, Verdesian Life Sciences has decided recently to put a hold on the project. There will not be any material available for sale to the public, as well as to state or federal agencies, and even to researchers this fall. They are, though, very much interested in gathering any data regarding the efficacy of this particular strain. The registration process of HCK55 is proceeding, but the soonest the registration is anticipated will be the first quarter of 2018, which would be well beyond the ideal application period for this particular biopesticide. Regarding MB906, the latest word from BioWest Ag Solution is that they are moving towards the preparation of a registration package for submission to EPA for the registration of that biopesticide. There we go, Matt.
Thanks, Rick. Can we shift the mic to Mike Gregg?
Thanks, Matt. Well, I'm going to spend my time talking more about the biology of this organism, how it works, and kind of some of the key questions that we have to address as we move forward. Pseudomonas fluorescens, the D7 ACK55 that Rick described, are both native bacteria. They colonize in soil residue and roots. And now this is a cold weather bacteria. They survive well at low temperatures, and they're not competitive at warm temperatures. So during the summer and the warmer months, they go dormant during the hot, dry summer. And they do not suppress crops or native plants, and that is by design, because when Ann Kennedy discovered these and started working on them, she didn't really just go find a bacteria and try to make a biocontrol out of it. She actually saw the concept, the potential use of these organisms to suppress weeds, and actually through a trial and error process identified those that were specific. So the specificity is really by design. And these bacteria produce and deliver weed suppressive compounds to the weed root. Next slide. Next slide. Now, one, I think, important point here is that the bacteria doesn't kill the cheatgrass. There's no signs of visible lesions. There's no signs of pathogenicity. The cheatgrass that's impacted by these bacteria are actually healthy plants. They just turn out to be really small because the bacteria, these weed suppressive compounds produced by the bacteria, inhibit root cell elongation. You end up with just stunted roots and a stunted plant, interrupts tiller initiation, and essentially reduces seedling vigor, tiller and seed production, and overall you start eroding away at the seed bank because you're producing fewer seeds. So ecologically, it's a good match for biocontrol because essentially what we're doing here is kind of leveling the playing field where cheatgrass that are impacted by Pseudomonas are no longer having that early germination or taking up resources and space and out competing our native plants. So really kind of it's leveled the playing field. Next slide. So there are some key requirements for success. And one, obviously, the bacterium must survive. And that may be easier said than done. It is applied in the fall when it's cold, temperatures less than 50 degrees, and needs to be applied with moisture. So it is applied either as a liquid, where the bacteria is mixed in the liquid and is sprayed on the surface of the soil, or as a seed coat where it's drilled in with seed. So for applying it as a liquid, you have a fairly narrow window in the fall when you apply it, when it is cold, before it freezes up. And once you apply it, either you have to have rain within about 7 days, or you need to go apply another liquid application because the bacteria just sits on the soil around the vegetation, it dies. So it needs to be incorporated into the soil. Drilling the bacteria with seed essentially does that for you. It also must establish over winter. So once it is in the soil, it needs to build in populations. So you have bacteria to inhibit cheatgrass in the spring. And soil type as well can be important. Some of the heavier clay soils or those with high organic matter may bind the compounds and not be effective. We don't really know a lot about in terms of what soils potentially might work best and what ones will not. And obviously you need to have the desired plants to really go inhibit and reduce cheatgrass so you don't have your desired plants, your native vegetation coming in, then over time you're just going to end up with cheatgrass again. So this is a long-term process. This isn't something where you go out and spray, see immediate results, kill plants. It's much more longer time frame, where I believe Rick mentioned it's 2 to 5 years, where you apply the bacteria, builds up in population, it peaks years from 2, 3, 4, and by 5 it starts decreasing again. So it's more of a long term. And the other important point is that the bacteria works on the cheatgrass that's just starting to germinate. If you go spray bacteria on the existing stand of cheatgrass that's greening up in the fall or in the spring, it's not going to do anything. This is one that impacts as those cheatgrass are germinating. Next slide. So there has been a lot of trials, and has done a lot of work. A lot of other people's put bacteria on the ground. There has been a lot of mixed evidence and efficacy. And so I started working on this probably 5-6 years ago. So I saw the potential for this in terms of use on rangelands, dealing with cheatgrass and sage-grouse habitat. But there has been, what I saw, there was a real need for a coordination effort. There's a lot of work being done, but none of it was really coordinated, where we could start using some of these trials and start learning and advancing our knowledge in a coordinated way. So the Fish and Wildlife Service sponsored a workshop in August 2015 that was designed to bring in, it was an experts workshop to bring in the folks to talk about design methods and implementation and where we really need to go with this as we try to scale it up and taking the science and actually developing a new management tool. So one of the key questions, obviously, is efficacy. And based on this workshop, we decided that pretty much, one of the first things we need to start seeing if we can actually scale this thing up and use it at a larger scale. So in the greenhouse or in 1 meter square plots, those sorts of things we can really control and do things right. You can see results. But if you start scaling that up, I think it's going to get more difficult. One of the other important parts for the non-target impacts, particularly with the soil microbial community, there hasn't not been a lot done work with that. Different soils, ecological sites that I talked earlier, the clayey soils tend to bind that toxin, so we need to really identify, what areas where it works and where it might not work. Delivery systems is another one. Like I mentioned, right now we have seed coating and liquid. Now the liquid application is the primary one that's used, but the D7 label recommends like 30 to 50 gallons an acre. So you're going to start taking that and put it on an airplane. That's a lot of weight. It's just at this point, I think, before this is really will become a large-scale tool, we have to start looking at some of the different delivery mechanisms. Maybe even a pellet form or granulized form that you can apply on the ground when it rains. Bacteria is washed into the soil. Those sorts of things where you don't have this narrow window, this limitation. We're actually trying to spray in November when you have freezing and thawing, and it just really makes it difficult for spraying equipment. As well as distance from the source material. This was all isolated in the Palouse prairies, and we're applying it in Utah and different places. Further we get away from that source, we really don't know what that does to efficacy. So I guess the point being that there are just a lot of questions out there, and I think, like Matt said, we kind of got the cart before the horse, and this was really starting to gain some steam, and people thinking that, well, as soon as this stuff is registered, we can put it on the ground and we'll be good to go, and we're just a long ways from that. And really need a coordinated research effort. And there are some of those things going on. Fish and Wildlife Service, myself and Dave Pyke are working on a project up at Hanford. That's a replicated study using D7 in really what I call a proactive and a reactive approach. The proactive is we're applying a liquid bacteria in an area where we have marginally invaded, where you still have some natives. So maybe we can inhibit that cheatgrass. Those natives will again start to take dominance. You can get that area back in or headed in the right trajectory. And more of a reactive approach, and that's post-fire rehab, where you are actually going out and doing some revegetation so you can incorporate this bacteria in with that and drill it into the ground with the seed. And we are in our second year in that. And like Rick's work, we don't have really any results that we can present at this point. So the next couple of years, we'll start having some results. I think with that, that is about all I have right now.
Great. Thanks, Mike. Can we turn the mic to Jane in Montana?
Thanks, Matt. So although we're not in the Great Basin, Montana does have its fair share of invasive annual grasses. So we've seen an increase in cheatgrass over the last 10 to 15 years. Next slide, please. And we also have a good amount of Bromus japonicus, or Japanese brome, especially in the eastern third of the state. Next slide. In 2013, we discovered our first infestation of Medusahead in northwestern Montana. And in this area, Medusahead is actually growing with cheatgrass, Japanese brome, and Ventonata dubia. Next slide. In the past 3 to 5 years, Ventonata has been on the increase in western Montana, as shown in this picture taken earlier this summer. Next slide. Cheatgrass and Japanese brome are also weedy in crop fields in Montana, particularly winter wheat. However, they seem to be more easily managed than in the rangeland systems, and this is typically done during the fallow cropping phase. Next slide. As for management on rangeland, many people are fortunately still in the position of preventing invasion. The photos I showed you in the previous slides were really, worst case scenarios in Montana. So many people are just managing small infestations, typically using chemical control. For more severe infestations, some people are trying grazing and even revegetation. But all in all, I'd say that there are a lot of people who are just not managing invasive annual grasses at all on their rangeland, somewhat due to the fact that they're not quite sure how to approach it. So with that said, there's been a lot of interest in using Pseudomonas as a bioherbicide. Next slide. So we established what we're calling a coordinated distributed experiment to test the effects of Pseudomonas ACK55 on annual brome cover in Montana and Wyoming. This project is being coordinated by Dr. Kurt Reinhardt, who's with ARS in Miles City, Montana. He has 3 sites in his area. There are 2 sites near Missoula, 1 site near Lolo, one site near Norris, and 1 site in eastern Wyoming near Bill. So this slide gives you a general idea of where the sites are and the people in charge of each site. Next slide. We applied ACK55 onto 5 meter by 5 meter plots, and each treated plot was paired with a non-treated plot of the same size. Each of our 8 sites has anywhere from 4 to 8 replications, and we had 1/10 of a gram of freeze-dried ACK55. That was mixed with deionized water in a handheld sprayer and applied in late fall or early winter of 2014. And these were protocols shared with us from Dr. Kennedy. So each summer since then, we've been sampling the percent cover of annual bromes or cheatgrass and Japanese brome in 1 meter square frames. So to estimate cover and keep our estimation process similar across the sites, we divided our square meter into 100 cells. And if an annual brome even occurs in a cell, the cell gets counted. So a cover value of 72 would mean that annual grass occurred in 72 of the 100 cells. So it's a little bit different than your conventional canopy cover of, for example, 72%. So our plots have been sampled 2015, 2016, and 2017, so we have 3 years of post-application data. So our preliminary interpretation of our data is that we're not seeing a large impact of ACK55 at our sites in the northern Great Plains or the middle Rocky Mountain ecoregions. We will be monitoring these plots through 2019, so that will be 5 years post-application. So I guess stay tuned for more outcomes. Thanks, Matt.
Thanks, Jane. And now for our last presenter, Bren Lazarus here. Brynne.
Thanks, Matt. So my role in this project has really been just to help gather information about the use of weed-suppressive bacteria across the Western U.S. and to connect the researchers and managers working with it so they can exchange ideas and share results. And there have been a lot of manager trials, either at the demonstration or the operational scale, that we can and we really should be learning from, along with some formal studies by researchers. So, but what makes a complete study, or what is needed in order to learn from a management trial? 3 elements: controls, replication, and monitoring. And I'll talk about each of these 3 elements, and then at the same time, give some examples from studies or manager trials here in Idaho. So by controls, I mean comparable untreated areas. So scientific studies generally have controls built into them, but managers usually need to get creative if they're going to include control plots, perhaps looking just outside their treated areas, or maybe temporarily turning off an ATV or a helicopter sprayer to create some untreated gaps within a larger treatment area. So this is an important point. If multiple types of treatments are combined, say herbicide and bacteria, then a complete set of controls must include each individual treatment type alone, in addition to those completely untreated areas. So I'd like to give you an example of how USGS worked with Idaho Fish and Game to get a complete set of controls into a large-scale management operation. So after a 2016 wildfire, Krista Byrne at the Boise River Wildlife Management Area notified us of her plans to treat 1,200 acres with tank mix MB906 and imazapic via helicopter. So we were able to work with Fish and Game to spread 3 tarps along each of 6 pre-existing vegetation transects to block the combined spray. After spraying, we removed the tarps and sprayed 1 in each set with MB906 only, another one with a Mazapic only, and we left the third untreated. So this photo on the lower right shows an unsprayed plot within the Mazapic plus MB906 matrix. Another critical element needed to make it possible to learn from studies and manager trials is replication, by which I mean treating and leaving controls for both multiple sites and from multiple plots within a site. So replication is needed to establish repeatability and also to characterize variability. No treatment has a perfectly consistent effect, and knowing the variability in response is important for managers. If I want to compare a treatment against a control, I need a measure of variability for each to help me understand whether the difference between them is significant or not. So I'd like to share an example of some fantastic within-site replication for a manager trial at the Tex Creek Wildlife Management Area in southeastern Idaho. After a wildfire burned the area in the summer of 2016, Fish and Game sprayed 6,800 acres with MB906 in multiple non-contiguous patches, the pink cheatgrass treatment patches on this map. They then stripped in herbicide, Mazapic, in 90-foot wide strips like the ones shown on the lower map. And they did that in 8 different locations that were treated with MB906 and then also in another 8 untreated locations. So at each of these 16 locations, they monitored cover in paired herbicide, no herbicide plots, which are represented by the 1s and 0s on this map, resulting in 8 replicates of each of the 4 treatment combinations. So Matt and I have enjoyed consulting with these guys, and we think their efforts to learn from their treatments are exemplary and are going to be very informative. So the third critical element needed to learn from a study or management trial is monitoring. Results must be quantified if they are to be shared with other managers and researchers, and thereby contribute to our collective knowledge. So one of the most commonly measured variables is cover, the proportion of area covered by a given species. And Jane described one good method of measuring cover just now, and I'll briefly describe 2 others. So one is to take downward-facing photos of the ground surface with a camera mounted on a pole, and then use a free software program called Sample Point to quantify the area covered by each species. So with another method for measuring cover, line point intercept, LPI, a pin flag is dropped at a point, and all the species that it touches are recorded. And this is repeated many times, either within a frame or along a transect. And I've included the websites for a couple of useful monitoring resources on this slide on the right side. And this graph shows an example of cover data we collected in a study I'm leading with Matt and the Idaho Sage-Grouse Action Team, which was installed in the fall of 2016 in southwest Idaho. And this study includes many treatments. They're listed along the x-axis, 2 strains of Pseudomonas bacteria, D7 and MB906, 2 herbicides, Mazapic and rimsulfuron. It also includes tilling with a drill seeder to work the bacteria in. And this study is replicated at 3 sites with 3 replicates per treatment at each site. And the graph shows cheatgrass cover in late spring of the first year after treatment at one site. So untreated plots and those treated only with bacteria have cheatgrass cover around 80%, while plots treated with herbicide have cheatgrass cover of 20% or less. I should emphasize, though, that this is preliminary year 1 data for one site only. So stay tuned, please, for updates from this project. And now I'd like to give you just a very brief overview of our findings to date from the Weed Suppressive Bacteria Coordination Project that includes over 24 participants since we started it this past summer. So we've tallied 34 manager trials, and these cover more than 30,000 acres and occur throughout the western US. With respect to the 3 elements, controls, replication, and monitoring, we can only confirm that 16 of these trials definitely include some comparable areas not treated with weed suppressive bacteria, but it's possible that there could be more than this. 14 of these trials combined herbicide with bacteria, but only about half of them also separated these treatments out individually. And only 5 of these have replication within sites, but if we look at the many trials and consider them collectively, these do comprise a measure of among site replication. And 22 are doing some kind of quantitative monitoring. Just a few, though, have all 3 elements. We've also tallied 14 scientific studies, which do have controls, replication, and monitoring. And this table shows that the majority of studies have focused on D7, with only 2 including MB906, while there have been at least 13 manager trials with MB906. And this figure shows the number of studies, those are in blue, or management trials in red on the y-axis for each time period along the x-axis, And you can see that the number of manager trials has boomed in the past 2 years, reflecting the recent commercial availability of MB906. However, despite the fact that 7 studies were completed between 2001 and 2013, there exists to date only 1 peer-reviewed publication on the effects of weed-suppressive bacteria in rangeland ecosystems. So we're really hoping to see more published information in the next several years. And finally, just a plug for this project, we know that there must be more manager trials and maybe even a study or 2 that we haven't yet tallied. And we'd encourage you to please get in touch with us, get connected to the community of other researchers and managers who are working with weed-suppressive bacteria.
Thank you, Brynne. Okay, now just to recap here and get us ready for some questions, we're hoping that this webinar helps everybody become aware of the situation of an emerging, largely unvetted and untested technology that is affecting how managers are allocating their resources for control of exotic annuals. And I think one of the most exciting things here is that we are able to coordinate. And right now it's on the topic of weed-suppressive bacteria, but as we all know, working in these ecosystems, every 5 or 10 years, there's some new stressor or tool on the table that needs to be dealt with. And we're hoping to demonstrate how we're trying to, again, get the horse before the cart to be proactive, and so that we're dealing, so that we're not faced with dealing with our problems in a reactive fashion. Our problems, again, could be things that come up from tools that we use without proper vetting. And I really like how we've got managers and scientists working together here. I think that's the way of the future as research funds continue to dwindle. The treatments that our land managers are applying at vast scales across the Great Basin and the neighboring ecoregions are the treatments that we need to be learning from. So thank you very much, everybody, for tuning in thus far. It's time to see if we can entertain some questions. Liz or Emma, do you have any so far?
Yeah, Matt, just to start off with, there was one quick clarifying question that came from Richard's portion of the presentation when he was giving the history. And I believe it was on slide 15. There's just a note on there that the EPA granted registration of the D7 strain. And the date for that was August 29th, 2017, which is tomorrow. I think there was just some confusion what that date actually was supposed to be.
I'm sorry, I couldn't hear your question completely. You wanted to know when the D7 was registered.
When did EPA grant registration of the D7 strain?
10 months after it was submitted.
Okay, great. That just came up during yours. I think there was a question.
Hey, Liz.
Gotta go back to 15. Go back to 15. Granted, yeah, that should be 15.
2015.
Hmm?
Great.
Thank you.
We're getting quite a bit of feedback from your phone line. Matt, can you confirm if you can hear me better by chance? Or if that
Correct.
feedback is just happening on my end?
Correct.
Okay, then Liz, if it's okay with you, I might run through the rest of the questions. We were struggling to hear you at the end there.
Okay, yeah, please go ahead.
Okay, so we had a question about, it says, is anyone from ARS responding to EPA's request for ACK 55 now that Ann Kennedy is retired?
As far as completing the project, yes, there is. There was an individual that was identified. I can't think of his name, but he is the one that has been working with EPA to make sure that the necessary studies that EPA requested are done and have been submitted to the EPA. I don't know if there is research being done. I guess I would have to turn that over to Matt, maybe, if he has any idea. But as far as the overall project of getting the ACK 55 registered, that is moving forward.
Oh, yeah. So there's a soil scientist up in Pullman who apparently is getting involved. I also knew that the ARS in Reno had been involved in helping get some of the tests, particularly the honeybee sensitivity tests, underway. So, but the good news is that there's action moving forward, and the ARS continues to be involved, although not Ann Kennedy.
Okay, thank you. The next question that came in was, why wasn't there a demonstration test of the products done in Nevada? With the fires, there are huge monocultures of cheatgrass available for testing there.
Now, is that a question pertaining to BLM or to just in general? If it's as far as the BLM is concerned, the field offices were given the opportunity to respond, and at the time we were putting this together, we didn't have a response from a field office in Nevada.
Okay, and I think that question did come in during that portion of the presentation. There's a lot of questions coming in, and I know we're limited on time, so I'm going to move on. There's some confusion about why herbicides are coupled with weed suppressive bacteria applications, and could someone please clarify why both were used for some trials?
That's a hard answer to generalize, and it really comes when managers are doing a trial, they are the ones who are deciding on how to devise something to their benefit. In other words, something that will work best on their landscapes. My sense is that herbicides are such a tried and true control agent that they feel that any tool or cocktail of agents would likely include a masopic. From a scientific perspective, it's a very complicating factor. Rick?
Typically where we understand, and Mike can fill in, but it takes approximately 18 months to 2 years through our understanding for the bacteria to build up in sufficient populations to where we'll see more of an efficacy. So we're using the herbicide. A 3-ounce rate of a masopic is not going to give you 2 years worth of control. It will give you 1 year of control. So in essence, you're kind of helping the bacteria get established by reducing some of the downy brome, and then letting the Pseudomonas build up its populations. Later on, the herbicide will have broken down and won't be a factor in the control.
Yeah, that's the point I was going to make. If you're going in with a fall application of bacteria and you have pretty good green up of cheatgrass there, the bacteria won't impact that growing cheatgrass. Use a glyphosate application to eliminate or take out that existing cheatgrass, which reduce seed production and that. So,
and another point to add to that is that it has actually been the recommendation of the manufacturer, particularly in the case of MB906, which is marketed not as a bioherbicide but as a soil amendment, that herbicide be co-applied with the bacteria. That's the recommendation of the manufacturer, at least in that case.
It's intriguing, though, because you'd think that these two agents would potentially have opposing effects. If the herbicide takes out the cheatgrass or other exotic annual, then what does the bacteria have to grow on for a substrate? So there's a number of issues involved with this. For now, though, what we're hoping people will do is, if they feel compelled to co-apply herbicides with the bacteria, that they separate out the treatments, at least on a few side test plots.
All right. We have several other questions that have come in. We have hit our scheduled hour time. If the speakers are able to stay online, we can continue to run through these questions. Does that work for you, Matt, and others?
Yes, it does. Yep. And we should also point out. We should also point out too that we've got very high enrollment on this webinar. Probably there'll be a few questions that we can't get to, but we'll have those questions written and we know who submitted them and we can respond via email also.
Definitely. Okay, well, the next one that came in was, what NEPA challenges to application do you expect and what is being done to address those challenges?
Rick?
Well, one of the things that that drives some of the research and demonstration is the NEPA and other issues. So when we put this together, we set it up so as to be able to address the NEPA issue in a small scale, so that we could, in our protocol, we would structure the protocol such that the identification of risk was minimized. And in working with the folks back in DC, we were able to use the information that is provided by the manufacturer to EPA to have a better understanding of what some of the potential risks might be. And so the fact that we were only treating no more than 50 acres in a field office at one time, we were selectively selecting sites that had less potential for risk. We were able to minimize risk. But yes, NEPA will be a factor that will be have to be dealt with and addressed as we move forward. If Rhodesian moves forward with D7, we don't know the future of the D7 product at this point in time. The ACK55, it's similar. We need to have more studies that tell us just how efficacious it can be, because it will be a driving factor. The NEPA process will be a driver.
Thank you. Okay, the next question was, are studies also working to test alternative application conditions, for example, date, temperature, weather conditions, etc.? And also a second question from this person, if a study finds no significant effect, how do we know it wasn't an application problem?
That is a huge challenge here. Yeah, that, and the short answer is each study brings in a few additional factors on application style. One significant researcher, Dan Takala from University of Wyoming, for example, is one of the only people that is adjusting application rates, like how much he adds. There are a number of studies which have reapplied. So they've tried applying once and then coming back the next year and adding more. Brynne, are there any studies that have modified, that have used season of application as an experimental factor?
I think Chad Boyd, I'm not remembering now if it's his Orovada, California, or his Harney County, but Chad Boyd from ARS has done some work with that. And well, actually some of the BLM trials that Rick's shown us, some of those are spring applications and some of those are fall applications. So there's a little bit of, they're playing with that a little bit with the BLM as well. That's all that I can think of off the top of my head, looking at season of application.
Yep, and then the other permutations are either spraying the soil, the bacterial agents on the soil surface, or as Gregg talked about, applying them onto seeds and drilling the seeds into the ground, or as we're doing here in Idaho, having a factor in which we till the bacteria into the soil. Those are probably some of the bigger ticket issues to hit first, but clearly the research that's ongoing now is really just going to scratch the surface, and it will most definitely need to be follow-up waves, like years of additional study try to figure out how to work with this potential biocontrol. Many questions that won't be answered in the near future. Now we're just trying to find some effect. Dave?
Yeah, let me add to that. You know, if we were to do a research study where we were going to address that aspect of it, and I've always been concerned about this, we really need to be taking samples of the material at the time that is being sprayed and actually having it analyzed for viable bacteria within the application. And, you know, we're not really geared up for that. We haven't done that, and especially in the field trials that are being done. This is a real concern that we have because we don't know where in the process things might go wrong. You've got maybe viable material that's coming from the supplier, but then, you know, is the handling of it as we go through the process of actually getting it sprayed on the landscape, could that be impacting it? And so actually working with some microbiologists who could actually determine that for us would be good to do, but that's a considerable expense that we're not geared up for at this point.
That's a good point. One of the biggest problems we have is that there are no molecular markers that can be used to identify the abundance, or the presence or the abundance of these strains of Pseudomonas in soil. That's a huge problem. So some of the existing studies right now are at least archiving frozen soil samples in -80 Celsius freezers in hopes that some lab somewhere will be able to create a marker. At least we can go back in time and see whether or not we even increased the you know, the Pseudomonas Fluorescens strain populations in the soil. So that's a huge black box for us right now. It's problematic.
Okay, thank you. The next question is, it appears that the 2 strains of weed suppressive bacteria will not work in the warmer desert Southwest. However, one of the products mentioned was an active ingredient. How does this product appear to work in warmer climates?
Active ingredient being the liquid formulation, I think.
It doesn't specify.
Yeah. Brynne?
I don't think we know that yet for sure. I think, so it's been really interesting. In my role as coordinator, I've gotten to call a bunch of people and say, hey, are you using this and how's it working for you? And really the answers that I get vary so much from, yeah, this is great, to, hey, this didn't work at all. And so the only way everybody can be right is if, you know, if there's some pretty specific application conditions or temperature, you know, weather, soil conditions that are needed in order for this to work. And so getting to a place where we can find that out is the challenge.
In warm deserts, it may be very difficult to predict and therefore plan and implement an application of this bacteria. I mean, it really, even here in the cold desert of Boise, we found it to be a challenge to try to time the application of the bacteria to get it in the soil during the specified cool, wet conditions. So the windows of opportunity will be scarcer in the warmer deserts. And by the way, I guess they would be scarcer in a warmer climate with, you know, potential climate shifts.
Great, thank you. Next question is, has mowing of cheatgrass prior to application had a trial yet?
Yes, it has. We know of one conducted on a Department of Defense installation here in Idaho. They compared mowing pre-treatment with pre-burning treatment. And what was the third? Do you remember? Those are two.
No.
And they there wasn't any notable results from pre-mowing. Although these the investigators think that pre-burning did actually stimulate a response, but whether or not their controls are appropriate is a question.
Thank you. So the next one is that Mike said that D7 stunted the growth of cheatgrass. Does it still set viable seed?
Mike?
Seed, my understanding, yes, it still produces seed and will produce viable seed. It just doesn't produce as many tillers as much of it because the plants are just much smaller and less vigorous.
Okay, thank you. Moving on to the next one, it says, curious about the status of the test sites for the D7 or ACK55. Specifically, were the test locations previously disturbed or were they active range/croplands, etc.? Has anyone looked at disturbing the location, tilling, roughening the soil up?
Yes, there's two different ways to go about this. One is, so the primary network of demonstration sites, the most cohesive one is in the BLM, and I'll ask Rick to talk about that in a moment. But before that, Brynne and I can tell you about the broader set of trials that have gone on by many different agencies. And by and large, most of the sites that are being treated have been disturbed in some ways, in non-planned ways. In other words, there's sites that were burned by wildfire, for example, within the last 10 or so years, and so they're already in a compromised condition. But many of the applications that have occurred have been immediately after either unplanned or planned fire. In other words, some prescribed burning. And there's a number of trials that also incorporated some sort of rangeland drill treatment. So there's not a simple answer to your question, except to say that yes, the majority, but not all, of the sites have been previously impacted in some way. Rick, is there anything you can summarize about the BLM network of demonstration sites?
General statements are, in visiting with some of the folks involved with it, yes, some of the sites have been burned. Some were burned within a year or two, others several years back. There was one site that had the soil roughed up, but it wasn't as part of the demonstration. This was the area where they wanted to put it. So it was not part of the protocol that we sent out to the field offices that the site be disturbed, but it's just part of the additional information we're getting from the field as they go back, as we start talking about what is the data saying, because like we've heard, the information that we get, the time of application, and the conditions, that may be what tells us what works and what doesn't work. So again, this is just a demonstration as far as the BLM is concerned. We're not researchers. I was most appreciative that the field offices expressed interest in working with the Verdesian Life Science folks to get these on the ground so that we could get a feel for what might be happening. And as we gather the data, I think we will be able to tell a better story of what's going on.
These are great questions because disturbances will affect the amount of litter and standing plant biomass, which affects how well, for example, a spraying of the bacteria could actually contact the soil. And then disturbances also affect whether or not you have a lot of young plants on the landscape, both the target as well as the non-target plants. And we think that the bacteria should probably be most effective on young establishing plants, which I guess you get every year with annuals. But just so everybody understands why those questions are being asked, it's for these reasons.
Thanks, Matt. One of the next ones was, how does the bacterium target annual grasses selectively, and what effects does it have on other plants?
That's the million-dollar question. That's what we all are dying to know.
But when Ann went through her sample, she went out and collected soil, isolated bacteria, and through trial and error and tests in the greenhouse, through process of elimination, the ones that were not specific, she gave no further consideration to. So she started with thousands, and then just essentially worked it out. And it was like 0.01% of the bacteria actually were something that could be used. So like I say, when I went through, she was actually looking for the specificity just through greenhouse studies and trial and error over the years to look for those bacteria that were specific to certain weeds.
Mm-hmm.
So it was by design. And then,
yeah.
So through that process, she was testing native plants, and she tested a lot of native plants. All of them, probably not, but so she has tried these bacteria in many of the native rangeland plants, grasses.
How it is that these Pseudomonas fluorescens strains could affect and inhibit the root growth of exotic annual grasses, but not seedlings of perennial native or introduced grasses, is somewhat of a mystery to me. And I do remember, Mike, at the 2015 meeting that Anne had some explanations. She did offer up a couple of explanations. She thought that the perennials and the annuals differed a little bit in their membrane structure, and I didn't see a lot of experimental evidence for it, but that's where she thought the anatomical difference might lie that would actually enable the specificity.
She did have an explanation of, like you say, the cell structures and that the cheatgrass compared to other plants were different, and that's how. So she did have an explanation for it.
Yeah, let me add too, Matt, that Harris in some of the early work back in the 1960s, I believe, but it certainly in the mid 1900s, did some morphology work on the roots of cheatgrass. And cheatgrass has a very weak, if not non-consistent Casparian strip. So that diagram that you've got there is typical of a perennial plant and many plants, but annuals and especially cheatgrass, at least there is some indication that waxy material that's associated with the Casparian strip, which tends to keep materials from passing across the membranes from the outside and keep out pathogens, that may be one of the reasons why a bacteria could work its way in.
Fascinating.
Thank you all. I'm going to move on to the next one, which was, could any of these agents be used as an inhibitor for areas of new surface disturbance in infested areas, or does it need to be applied in areas with active growth?
Well, does anybody have an answer to that amongst the speakers? I could offer one.
Well, the only thing that I can think of is that it does require to have the cheatgrass there, or that's my understanding. So the lack of cheatgrass entirely could potentially mean that you're not going to have a very quick increase in the bacteria itself. So it's not generally thought of as being a mechanism for using when you just have a small infestation. Although one of the areas that we're working on there in Hanford, and correct me if I'm wrong, Mike, my understanding was that we did spray an area that had native plants and cheatgrass within it, right?
Correct. That was the one where, we did nothing except spray the bacteria.
So we may have some information in the future, but it's one location.
Thank you. Okay, the next question, I think we'll do a couple more, and then we're going to leave some of the more specific questions, I think, for follow-up by email after the presentation. But this question is, Pseudomonas was isolated from naturally occurring populations in eastern Washington, I assume. Clearly, it is possible that there is variability in Pseudomonas across the continent. Is there any research that addresses variability in the species, presence of subspecies or other effective species, and potential for differential effectiveness based on origins of the cultivated bacteria?
Well, Brynne and I have probed the literature for that, and we can't find anything in soil, in the Pseudomonas fluorescens strains in soils that would help answer that question. And it's the question is a very big concern of ours, both in terms of making the tool effective, but then also for potential collateral impacts. It's a real problem. That's a glaring information gap.
Okay, and then I think we'll end on this question. It says, for those who are interested in implementing a project, do you have a survey form or questionnaire or any place you direct them where they could provide, or that you could provide, that might assist managers in design of their project to allow their results to be most useful for data analysis?
Mm-hmm. Yeah, I would highly encourage anybody interested to reach out to us. Bren Lazarus is actually receiving part-time salary support from the BLM to help in this very regard. So she, as well as myself, talk to managers a lot, and also now with this coordinated network of people that we have, we could very efficiently align different managers or researchers to others near them to help out, or to different specialists. So there's no documents available now aside from the manufacturer's guidelines, which we would recommend not relying on purely.
But I think that my presentation hits some very general ideas. Controls, replication, and monitoring are necessary, but that's not a bad idea to maybe come up with a more specific survey form. There isn't necessarily one right way to do any of this. But yeah,
a couple of things. There are a couple of things to keep in mind on this. As we've seen in this conversation today, there's a lot that we don't know. There's a lot that needs to be done in order to get a better handle on how it works, how the microorganisms, how it just behaves in the soil. I would like to see more replicated research done by universities, and there are programs in various states where the weed science researcher is doing studies to further understand how this behaves in the soil. Replicated work. The BLM were not replicating our projects. We're demonstrating. We wanted to see. Surprised us that there wasn't a lot of data, and now there is some work being gathered, some of the work that Matt's doing. There's researchers in Wyoming that are doing replicated studies to get a better handle on how this behaves. So I think part of it is, yes, it's nice to do demonstration work, but we really need to get the science out there to make it more efficacious and reproducible.
Mm-hmm.
Yeah, that is the take-home message of the talk. Excellent closing point.
Okay, and we will provide the rest of those questions, as you said, Matt. We'll provide them in written form to the speakers, and we'll make sure that responses get sent via email.
Great, thank you. And a virtual round of applause to our esteemed presenters today. Thank you all. And thank you to our audience. We appreciate your engagement and interest in this topic.
Thank you, Matt.
There's one closing slide to leave up. There's some emails that are available. Oops. Sorry. Emails are available on the website now with this presentation. And a recording of today's webinar will be available. And if you have any more questions about the Great Basin LCC, contact John Tull. His info is there. And please give us some feedback on the webinar when you log off. We use the feedback here to fine-tune our webinar series. Thanks.